Good afternoon, all the delegates. Welcome in session 2 of day 3 of 3 days international e-conference on pharmaceutical research and innovations to tackle future healthcare arena. I am extremely sorry, Dr. Hitendra Mahajan sir and Dr. Rakesh Tekade sir, for delay by 10 minutes in commencement of this session. I welcome Dr. Mahajan sir and Dr. Rakesh sir for this session. First of all, I will introduce session chair of the session, Dr. Hitesh S. Mahajan, working as Professor and Head Department of Pharmaceutics at R.C. Patel Institute of Pharmaceutical Education and Research, Shirpur. Sir has completed his M.Pharm from Government College of Pharmacy, Karad, and completed PhD from Government College of Pharmacy, Karad. Sir is having huge experience of 19 years in teaching and research. He served as course coordinator for certificate course in drug regulatory affairs and intellectual property rights, President Institute Innovation Council Cell IICC, coordinator NIRF and ARIIA. Dr. Hitendra guided 78 M.Pharm students and 3 PhD students, and 8 PhD students are pursuing their doctoral research under his able guidance. He delivered more than 28 guest lectures at FDP, STTP, national and international conferences to various colleges across the India. He received Excellent Teacher Award by Kavitri Bahinabai Chaudhary, North Maharashtra University, 2017-18. He elected as a board of member of studies for pharmaceutics by Kavitri Bahinabai Chaudhary, North Maharashtra University, Jalgaon. If we look after the research areas and research grants grabbed by Dr. Mahajan, completed sponsored projects as principal investigator, costing 1.22 crore rupees, national and international publications, 126 plus publications and with 2000 plus citations. 2 patents are granted on his name, authored 3 books and 2 book chapters, conference and papers presented in more than 50 conference and seminars and symposia. He is the editor-in-chief of International Journal of Pharmaceutical and Analytical Research, and he has been honored and been a recipient with many prestigious awards and recognitions for his credibility in the research work. I welcome Dr. Hitendra Mahajan sir in this session. Welcome, sir.
Thank you.
It is my great pleasure to introduce today's eminent speaker, Dr. Rakesh Tekade, working as Associate Professor, National Institute of Pharmaceutical Education and Research, Ahmedabad. Dr. Rakesh completed his PhD from Hari Singh Gaur Central University, Sagar, India. Pursued his Masters in Pharmaceutics from H.S. Gaur University, Sagar. Completed his graduation from Rajiv Gandhi Technical University, India. He is GATE qualified candidate with All India rank 1133. GATE was conducted by IIT New Delhi. If you look after the achievements, he is Commonwealth Fellow UK 2009, National Doctoral Fellow AICT, National Doctoral Fellowship, Irish Government Postdoctoral Award, CSIR Senior Research Fellowship, India and Junior Research Fellowship, AICT JRF Award, Fundamental Research Grant from Malaysian government with 1,33,000 Malaysian ringgit as a principal investigator. Experience, Dr. Rakesh is currently serving with NIPER Ahmedabad since February 2020 as an associate professor. He worked as assistant professor in IIT Jammu from July 2019 to January 2020. He served with NIPER Ahmedabad from July 16 to June 2019 and lecturer at Department of Pharmaceutical Technology, International Medical University, Malaysia from August 2014 to May 2016. Dr. Rakesh pursued his postdoctoral research. He was postdoctoral fellow at University of Texas Texas, USA from August 2013 to June 2014. He completed two postdoctoral research works. He was there at University of Hawaii at USA from July 2012 to August 2013. He was faculty of pharmaceutics at Sri Ravi Shankar College of Pharmacy, Rajiv Gandhi Technical University, Bhopal from 2011 to 2012. If you look after the publications, Dr. Rakesh has 300 plus publications on his credit, and if you look after the citations, it is about 6,800 plus citations with H index of 46 and I-10 index of 110. He is editorial in chief of five book and book chapters. Total patents are five, and two patents are under drafting with patent attorney. These are some snapshots of the books edited or completed by Dr. Rakesh Tekade. It includes dosage form design considerations, dosage form design parameters, those basic fundamentals of drug delivery, biomaterial and bionanotechnology, drug delivery system, the future of pharmaceutical product development and research, biopharmaceutics and pharmacokinetic considerations, pharmacokinetic and toxicokinetic considerations. Almost all books are from Apple Academic Press. With this, I welcome Dr. Rakesh Tekade for the talk on dendritic technology as the next generation siRNA delivery modality. I welcome Dr. Rakesh sir. Rakesh sir, over to you, please.
Thank you very much, Dr. Prashant, for this nice introduction. Is my slide visible, Dr. Prashant?
Yes, sir. Yes, sir. Your slides are visible. Please make it slideshow only. That's all, please.
Yes. So I had already made it in slideshow. Is it now in slideshow mode or not?
No, sir. You just make it once again, please.
Okay.
No, sir. You share it once again, please.
Yeah. Once again, I'm sharing. Yes.
Yes, sir.
How is it now? Is it okay?
Yes, sir. Yes, sir. But slideshow is remaining. Please make it
Oh,
slideshow.
uh, I made it in slideshow. I am unsure why it is not coming there. It's already in slideshow from my end.
No, sir. You have to share the screen instead of sharing window.
Okay. 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 Share screen. Okay, I got it. Now is it okay, Dr. Prashant?
Yes, sir. Yes, sir. Yes, sir. Slideshow is appearing. Please, sir. Please.
Okay.
Please.
Okay.
Please. All the
Excellent.
best, sir. Thank
Excellent.
you, sir.
Thank you. All right. So first of all, thank you for this nice introduction, Dr. Prashant. Uh, Before I proceed, let me take a quick moment to thank the organizer of this very valid, planned, and very thoughtful conference, I must say, International Conference on Pharmaceutical Research and Innovations to tackle future healthcare arena. The component which is in the title, that is to tackle the future healthcare arena, is very important and a hot topic of debate nowadays. And this is exactly where my today's presentation will focus around. So the title of my today's talk is Dendritic Technology as a Next Generation siRNA Delivery Modality. Let me take very quick moments to describe what are my objectives from today's presentation. With this presentation, basically, I want to crosstalk the importance of siRNA or RNA interference therapeutics. What are those? Why they are important? And why still our market or the clinicians have not realized this product effectively in spite of several years of research? So what is the ongoing problem, right? And what are the strategies that are available in the in resolving this problem, which are primarily related to the siRNA delivery, right? So what are the available options and what are the shortcomings associated with them, we will discuss in depth, right? Then Eventually, I am also interested in sharing the hybridized dendritic technology which is developed in our lab, and we'll also share some proof of concept data. I will try to make it as general as possible with least explanation of technicality of this project. And this project is funded by INSERV, right? And also eventually we got also grant from MOSTI Malaysia, Department of Pharmaceuticals, and of course NIPA Ahmedabad, our parent institute, has also significant stake as a funder for this project. We also got some funding from ICMR for this. So thank you very much in hand for all these funding agencies for their confidence in our research. All right, so before we move ahead, let me first describe you the central dogma of life. We all know that central dogma of life is a belief, is a set of belief, very well and loudly acknowledged. It is a concept that is basically, if you try to zoom the healthcare system, you will see that whether a person is healthy or unhealthy, the central dogma of life is the one that can clearly define it and crosstalk it. How? Let us try to first see that. So, how central dogma of life can define whether a person is healthy or not? Let us try to zoom the entire human body. Zoom the body, we'll get into some organs, the tissues, the cells. Now, this is the cell. We all know that cell is the structural and functional unit of the body. Cell comprises of nucleus. Nucleus comprises of chromosome. Chromosome got DNA. DNA got some functional units called gene. Gene got codons, which codes for mRNA by the process of transcription, and it is very well known. This mRNA get processed in the ribosome, which are the mini factories running inside our cells, right? So what they produce? They produce polypeptide. Why they produce polypeptide? They produce polypeptide because it got certain functions. It got certain functions, maybe contractile function, muscle movement function, nerve function, or storage function, hormonal function. For example, insulin is there. So the bottom line is, this gene if this gene is correct, if this gene is okay, this polypeptide should be okay. If this polypeptide is okay, the function what it is leading to is okay. And this is a time we can say that the body is a healthy human body. Let me try to put it in one simpler version. So what we have talked about is a regulated central dogma. Regulated means which is in control of the body. What's happening in regulated central dogma is a DNA producing a gene, very simple, which is DNA is having a gene which is normal, and by the process of transcription it is producing mRNA, which is producing polypeptide by the process of translation. And since it is a normal gene, normal mRNA and normal polypeptide has been produced, it is producing normal function, normal pathophysiology, everything is good. The individual is healthy, happy and fit. Yes, it is only because of the regulated central dogma. Now, consider the other side of the coin. A person is ailing. How central dogma is related? So that is called the dysregulated central dogma. It's not in the control. What's happening here? DNA is producing M gene. What is M gene? Is a mutated gene, right? The gene which has undergone some mutation. So this mutation can be because of any reasons, right? Maybe it is hereditary, maybe it is habit. The person is, say, sleeping late or eating unhealthy, smoking, drinking abnormally, ultimately leading to the production of M gene, mutated gene in the body. What it will lead to? It will either, of course, this mutated gene are also the gene functional, right? They will undergo transcription. They will also undergo transcription and produce mRNAs, which are abnormal in nature, right? And the polypeptide they produce will also be abnormal in nature. And this is the time we or the patient or the person start absorbing or feeling certain disturbed pathophysiology in the form of certain diseases, right? Say cancer or depression or retinopathy or diabetes, right? And this is the time we go to the doctor. And doctor, what he does, he gives us subjective treatment. Okay. So for example, in the disease, if he is weak, he will give you some nutrition. If the, for example, in cancer, if the cells are growing abnormally, he will give certain drugs, say vinclozine, vinblastine, melphalan, or methotrexate to control the cell division. If it is having the propensity of undergoing metastasis, it will give some drugs that will inhibit the metastasis, right? So treatment subjective. Treatment is subjective. That too with the help of certain API, the drugs, okay, which are our contemporary mode of treatment currently, right? So as per recent conferences that are being held across India, including the DOPs and several platforms, it was loudly said that we need to break the ice. We need to really go away from the conventional treatment approach and find certain alternative approach. Relying on one source, on relying on something particular is not a good idea. So in this context, RNAi therapeutics, the RNA interference therapeutics, they refers to an alternative therapeutic strategy. They are also regarded as next generational therapeutic modality. We will first cross talk about this RNAi. So this RNAi therapeutics, in context to it, 2006 Nobel Prize went to Sir Andrew Fryer and Craig Mello. We all know very well that it's for the physiology and medicine. So what is this RNA interference therapeutics? They works by how they work. They work by silencing the gene expression by neutralizing the targeted mRNA or protein. And what these genes are? These genes are the abnormal gene, the M gene. And this RNA therapy is a very, very potent. So if you talk about, say, some anti-cancer drug, their IC50 values usually in micromolar or nanomolar range if they are too potent. But in context to this RNAi therapeutics, okay, their IC50 lies in, say, picomolar range. They are very, very potent. Okay. There were several reports that suggest that if the, you might have heard in several instances, most of the time in movie also it said that now the drug has stopped working, dawa ne kaam karna khatm kar diya hai, now dua has to be done. So what is exactly happening in that stage? That is the point where drug has stopped working. That means the drug resistance has emerged medically. And it was found that if you couple RNAi therapeutics, strategically selected one with those same drug, then it will reverse the drug resistance. The same cell which were resistant are now sensitive towards the same drug. This is RNAi therapeutic. They are very simple in synthesis as against to the API synthesis, very well. They are not environmentally safe, right? And of course, these RNAi therapeutics are one of the drug alternative. Drug alternative means they can help us moving away from the conventional dependability on the API. This is a recent Times of India, and there are several platforms you can read this report that India are heavily reliant on foreign countries for their API requirement, especially in context to China, right? We are tremendously dependent upon China. Considering the ongoing geopolitical rearrangement that is taking place throughout the globe, this is very serious. You can imagine the situation when if these foreign countries or the exporters deny the supply of API to our country, what can be the scenario? It can produce medical emergency in our country. We will be in soup entirely. Our medical healthcare system will break down tremendously. Not only India, India is one of the biggest pharmacy of the world. The whole world will suffer, right? So what does this infers? This infers that we need to go away from this conventional approach, try to identify some alternative. So here again comes into the picture RNAi therapeutics. Very simple language. How they work? They work by replacing the mutated gene, by knocking it out and restoring the normal function. When I was in my first postdoc 10 years before in Hawaii, that was the first time I started working on this siRNA or RNAi interference therapeutics. And that was a time no um, such therapeutics were there in the market. But the good news is that in last 2 years brought these two product in the market. One is on Patro, which is for the treatment of polyneuropathy, and one is Givlari, which is for the treatment of acute hepatic porphyria, in which the toxic. So these are the two conditions, indications where the drugs are not available. So what happens in acute hepatic porphyria? Toxic porphyrin, aminolevulinic acids, or some neurotoxin, they get accumulated in the liver. So this Givlari, it is an RNAi therapeutics. It is given once a month for 6 months, and it was found that it reverses, clears all the neurotoxins, porphyrins, aminolevulinic acid, helps recovering the patient. Very potent. And the good news is that they are already FDA approved and in the market. They work by very simple process. Once entering inside the cell, they come in contact with agonite protein. This agonite protein directed towards forming a complex called RNA interfering silencing complex. This RNA interfering silencing complex, as soon as it forms, it gives the siRNA ability to identify the complementary sequence of the gene, which is actually our targeted M gene, mutated gene, and ultimately lead to its silencing. That is a very simpler mechanism by which it acts. So now, if you look into the literature, you can see that this RNA therapeutics, n number of clinical trials has been done, and most of the clinical trial, if you try to, classify it, most of them are can be tracked back to cancer. So most of the research in context to RNAi therapy is going around cancer, but it's not limited to cancer. But recent years have monitored and noticed its application for several diseases, including Parkinson's, retinopathy, diabetes, infectious diseases. So now it is all the way. Why? Because now it has identified that, as I said, we are currently trying to give symptomatic treatment. We are not actually targeting the root cause of the disease. Okay. And it was found that almost all the diseases, if you track back, zoom it, you can track to a master gene, which is actually producing all or leading to that disease. So, friends, it may possible that in the future, you go to the doctor and doctor give you a prescription having a pill comprising of siRNA. That's highly possible. We are heading towards this future. Whatever we have thought in the past, this generation is truly seeing that happening, including, say, nuclear powered machines, including electronic vehicles, artificial intelligence, Star Wars. So everything, whatever humans have thought once long back, is not taking into shape. And this is one such technology. I just said that currently our Medicare is trying to tackle the symptomatic or trying to offer symptomatic treatment for a disease. For example, let us take an very nice work by Sheena Itel. She reported that uh, she tried to identify exactly what happens in prostate cancer. Let us take an example of prostate cancer to identify why this gene therapy is important and how they are much, much, much better than that of the conventional drug therapy. So friends, this prostate cancer, what happens here? Let us try to see a very general picture. What happens here? Number one, Amplified glucose uptake, yes, it's happening. Cell division and uncontrolled proliferation is happening. Anti-apoptotic gene overexpression, and that's the reason they are not dying. High cell invasion, so that's the reason migration and metastasis is taking place. Yes, resistance is happening. So whatever drug you give, eventually it develops the resistance, and then the cell eventually attains immortality and stemness. So this is what happening. So now what currently oncologist is trying to fix at the moment is trying to stop the cell growth by giving certain cytotoxic drug, trying to give certain anti-apoptotic drug to combat this apoptosis, trying to give certain drug that can combat the migration or metastasis, or include certain drugs in the regimen of treatment of this cancer patient to avoid drug resistance. So that's the reason cancer therapy is basically a combination therapy, combination of several drugs. Agreed. Sheena et al. She identified that this glucose uptake is taking place because of this hexokinase overexpression, HK1. The SERT1, which is silencing mediating type regulatory gene, that is basically responsible for proliferation. Okay, and then this anti-apoptotic is because of overexpression of certain gene called CD44. And similarly, if you talk about metastasis, uh, it is because of cras1. Resistance is because of multi-drug resistant one gene. Immortality is because of this notch1. So if you want to tackle this, you need to individually give drugs that tackle them. But Sheena et al., what she identified that there is one microRNA 34. Actually, this is the root cause of the prostate cancer. When it goes up, it leads to production of HK1, Notch1, CD44, and down the line, this pathophysiological situation emerges. So, what's the take home message? The take home message is that if we want to treat prostate cancer, in spite of treating or tackling the anti-apoptotic pathway, or in spite of tackling the cell division or drug resistance, it's better and wise to tackle this microRNA 34. Why? Because it is a master switch. If you switch it off, this all pathways will switch off automatically. Better alternative, more effective alternative. Now, this is a I have tried to incorporate less of the data. So this is one from the same, right? That says here, this is the green one is the curve for the Taxol. It's a paclitaxel tubulin stabilizer. Its cell viability, the trend is this, and this is the one which is that of the MIR34. So these are very potent. Its IC50 are lying in very small level. They are required in very small concentration as compared to that of the Taxol. And this is one in Taxol resistant, that is paclitaxel resistant cell line. How they are being produced? They are being produced by gradually treating the cells with Taxol and developing a resistance state in them. So in them, further this IC50 goes up. In that also, this MIR34 is potent. And this is the one which is the IC50 value. We took it from here. So IC50 value of Taxol, which is marketed paclitaxel, right? 600 nanomole. In resistant cell line, it goes to almost 900 nanomole. While in case of MIR34, in fact, in resistant cell line, it is less than 100 nanomole. They are very potent therapeutics. Okay, this is the take home message from here. This suggests that if you treat this resistant cell line with Taxol, its IC50 is around 900. But when you combine this MIR34, say here 80 picomole, its IC50 come down to around 300. So what's happening here? It's happening because it is trying to sensitize the cell more than the individual chemotherapy. The next is why it is exactly reducing the IC50, because it is acting on the MDR1. From that previous slide, we got that this MIR34 is having direct impact on silencing MDR1. So what is it leading to? It is leading to this MIR34 is leading to silencing of MDR1. So multi-drug resistant gene gone down, the resistance gone down, and the sensitivity gone up. It's because of MIR34 gene. So this MIR34 again, how it is leading to anti-cancer effect is because it is signaling different cascade. Okay. Bottom line is that MIR34 is certainly a master gene. Okay. If you silence it, it will silence everything. Now the problem statement in front of us is very simpler. That silence the MIR34 gene and get rid of prostate cancer, but that's not happening. We knew this since 10 years, but nothing had happened. Why? That means there is a big in vitro and in vivo disconnect. What we mean by disconnect? By disconnect, we mean if you test this technology in cell culture level and you test it by say RT-PCR or Western gene expression profiling, toxicity test, MTT, XTT, you will get a very good data, good activity, good publication, and probably you will get good number of patents. But when you test in humans in clinical trial under in vivo settings, they are a big flop, less activity and less efficacy. What's the reason? The reason for this in vitro in vivo disconnect is a short half-life, it's a rapid renal clearance in 15 minutes. It's an in vitro in vivo degradation. Our blood comprises of a RNase enzyme. It basically it lyses it. They are polyanionic in nature, that is negatively in charge. Our cell membrane is negatively charged. Negative do not like each other. That's the reason low transfection efficiency. Even though if you flush the body with exceptionally higher level of siRNA, what happens? Maybe some may reach into the endosome. What happens there? It may trap, it gets trapped. So in order to have its effect, they should be available in the cytosolic compartment of the cell. That's a big problem. That's not happening. Another is off target effect. They don't know where to go. If they go to some other parts, a brain, liver, lung, kidney, on top of prostate, they will do this silencing there itself, leading to off target effect, toxicity issues. What's a lead? That means we are in short of an effective fit to purpose transfecting carrier system. Who will carry this gene therapeutics to the cell and deliver in the cytosol? That's a big problem where the research is ongoing. So we need some carrier that can do transfection. What is a transfection? It is a process of deliberately introducing this genetic material we call transfectant like RNA therapeutics inside the cell. There are several available. As I said, years of research. Optoporation where laser is being used to drill some micropores in the cells to push this RNA therapeutics inside the cell or form certain complex with calcium phosphate, and this complex having higher transfection capabilities, a micro injection, electroporation, and lipofectamine. On top of it, some viral vectors have also been developed, but they all are allied with certain problem. For example, if you take about viral vectors, they are toxic, immunogenic, and one more thing, if you use this, then viral vectors, they have a property, they get integrated in the genome of the patient. So what will happen? Reversal is not possible. So it's not just if you cut down the administration, the effect will cease. It will keep on going. That's a problem. Again, the problem statement is same. We are in need of in vivo reliable transfecting reagent. That's a big thirst area of research. Okay, we have decided in collaboration with biotechnology department, okay, let's try to find certain alternatives. Let's try to find some solutions. There must be some checkpoints, formulation lead, ki karna kya hai? What's the problem that need to be tackled? first thing that comes in our picture is that we are dealing with some moieties that are negatively charged. So you need some arrangements that can load this negatively charged therapeutics. One more thing to keep in mind that they are nanomolar concentration active. So their IC50 is very, very small. You cannot afford to have multi-step formulation strategy because they are required in very, very picomolar range. So what you need is a very simpler strategy for loading of this therapeutics. That should be one step mix and deliver type. Okay. And then we want something that can deliver it in vivo. Delivering in vivo is not sufficient. It should remain stable there. As we have discussed, RNase enzyme are there. If they are freely accessible to RNase enzyme, it will lead to their degradation. Okay. They are successfully inside the blood vessels. Not enough. They should permeate there, from there to the tumor site. We are taking an example of tumor. Not only the tumor site, they should go inside the tumor. Not only that, they should release it in the cytoplasm, cytosolic compartment. Okay, what's available in the market? In the market is, it's a very big market. I'm happy to share that this market is growing at an CAGR growth rate of 7.7%, which is nothing but exceptional. Its current transfaction technology market is going to be around 1.5 billion by 2025. This is really a big market, and this I have taken from the Mordor Intelligence or markets and markets.com. Currently it is 9,000 USD. Okay, so this is a big market, and out of this, most widely used one is lipofectamine, which has been launched like ages before 1993. Come on, it's still in use. It's having again a very big market and very big demand too. It's 1.5 ml vial. This you can see here. It is costing around 445,000. That is equivalent to around 7,000 USD for just 1.5 mil. It's having a global market of 351 million USD. But again, this is not perfect. They are good when you talk about the in vitro. Low stability is one that is associated with it. Why? Because it get unfolds. It doesn't have anything that makes it a proper vesicular structure. It lacks specificity. It doesn't have any targeting arrangement, so it's not specific to the target site. Low entrapment is another one. Offers incomplete RNS protection. It's not bad, but it's not perfect RNS protection. And one big concern is too costly. It's 1.5 mil and 45,000. That's a lot. You can imagine if anyone wants to develop some treatment formulation with this, how costlier it will be. So in our proposal, we have identified hyaluronic acid as it is an FDA approved polymer with CD44 analog, which is very specific for cancer. Okay, we've got some functional group that allow its surface modification. So we have identified hyaluronic acid because they are CD44 receptor analog. So that means if you formulate anything, if you incorporate this hyaluronic acid in your formulation architecture, then it is going to get selectively uptaken by the cancer cells. This is one thing which is very positive in its side. It got some modifiable functional group which are available. So problem with this is that they are negatively charged. Carboxy is negative and the siRNA which we wanted to deliver is also negative. So it's not possible to load it inside. So the first thing that one wanted to create is to catalyze it, modify, produce a new version of this hyaluronic acid, and then followed by this, as I said, this RNAi are very potent, so we cannot afford a multi-step loading and formulation strategy. So in our work we have developed and patented a never technology which can just mix and deliver type. So that means if you we have optimized the protocol in such a fashion that even a doctor clinician without any formulation expertise just can take reagent A, can mix it with B, add C, and then inject it. So we have done a lot of optimization here. I'm not going into detail, but we have formulated a simple polymeric architecture for the delivery, which we have called oncofectamine. Onco because it is related to cancer. Factamine means because it is having some dendritic amine into it. Okay, there is a strategic uh, reason behind incorporating or dendronizing it with dendrimer. We have dendronized it with dendrimer because it is a pH sensitive. This we have observed during 2009 um, when I was in Professor NKJN lab, that these dendrimers, they have a unique property of protonation when they get exposed to acidic environment, and we all know that cancer is having an acidic environment, so that's a very big plus. Plus the another problem, that is entrapment inside the endosome. Endosome is also acidic in nature, so there it can get protonated. You can see here, as it goes towards the acidic environment, it start bulging in size. This is called the protonation, proton sponge effect. So this acidic environment, the primary amine, they get protonated. As they get protonated, this primary amine, they repel each other, ultimately leading to increase in its size, creating a positive pressure on the wall of the endosome, leading to its rupture and releasing this loaded RNA therapeutics in the cytosol. And that's a strategic involvement which we have done in this work. Our objective here was to, if you see the literature, people have recommended using EDA, ethylene diamine, for cationization. But here we went away from that conventional approach. Here you can see if you use this 65 molar or moles of EDA, it will lead to production of around 40% enhancement in its amine architecture. But if you use this dendronized technology, it will lead to closer to 250%. So it is like several fold effective. So it produces very effective, very potent uh, dendronized polymer. Here, if you see, the reason for its protonation is because if you put it under acidic environment, it get protonated. Its zeta potential goes from -25 all the way towards positivity. So that means more and more amine groups are getting positive charge, protonated, repelling each other, and that's the reason it undergo swelling. They are very safe because we have defined it and developed it for in vivo administration in the in vivo environment. So they should be safe towards the blood. And here we found that they are pretty much safe as compared to that of the paclitaxel, which is like around 20% hemolytic. They are less than 1%. They are very safe, don't have any toxicity by itself. And this is the one which we have patented. We have developed three reagent. One is the oncofectamine, the other one is the dissolvating reagent. We have optimized several. So basically our work comprises of three reagent one form the complex with that of the RNAi therapeutics, followed by this followed by this followed by this add reagent 2, which is nothing but the one that can form that can convert it into nano architecture in one step. Just take one reagent 1, add RNA therapeutics to it. We have optimized how much maximum it can load, so there is a range how much max you can add. Okay, the same comes with the lipofectamine as well. Add reagent 2, just mix it in a pin off tube. Okay, in a tube, small tube, 2 ml is fine. Shake it, and then it will convert it into a micro nano vesicles. Okay, then add reagent 3, which we have again optimized. It is a stabilizing agent so that it doesn't get unfolded one. If it unfold, then what will happen? Then again there is no point of loading it, because eventually after moving in the dynamic in vivo circumstances, if it is unfolding, again it is making it accessible to the um, RNase enzyme, leading to again failure of your dose. So that's a reagent 3. And effectively it forms nano vesicles of size, nanoparticles of size, you can say, uh, around 280 nanometer, where reproducibly you can produce it. And this is very interesting, which we have observed. If you incubate this nanoparticle in acidic environment, their size become gradually towering, increasing in size with acidic environment, and that's good. So that means if you inject it in blood, it will be say of 280 nanometer. When it goes to the tumor site in endosome, the size is like several fold higher. Okay, eventually increasing the pressure inside the endosome, leading to the rupture of the endosomal wall. This is exactly what we wanted. We have also tested it by loading it, loading certain dyes, FITC we loaded, and we found that if you put it inside the acidic environment, then it will release the drug faster. Why? Because now the protonation has happened. Because of the protonation, the crevices, the loading crevices inside the oncofectamine has increased, and that's the reason it can go flawlessly, effortlessly out of the architecture. Okay, this is a very important component. That is the stability in the in vivo circumstances, and we all know that it is getting degraded in the serum is because of the RNAs. So what we did, we incubated the free siRNA without anything, stable until 24 hours. But when we incubated with serum, within before 30 minutes it got degraded. Lipofectamine was found something that can partially stabilize it, but with this technology you can stabilize it significantly for a prolonged period of time. So they are pretty much effective in protecting them from the RNAs. Then our proposition was that they are that the nanoparticle will get uptaken inside the cell by receptor mediated endocytosis following CD44 receptor. So what we did here, we first blocked the cells CD44 receptor using hyaluronic acid, and then we found that it reduces their cellular uptake. What does that mean? That means that the prominent pathway by this followed by this nanoparticle is CD44 receptor mediated endocytosis. And after pre-blocking the CD44 receptor, what's happening? After blocking it, their cellular uptake has gone down. Okay. So this infers that the primary mode of cellular internalization was CD44 mediated endocytosis. This is a gene knockdown, which we found that oncofectamine was much better. And then we also tested the cell viability, where it was found very effective in MDMA231. We have published our outcomes in scientific reports in molecular pharmaceutics. I welcome you all to read the paper and discuss if we can be of any clarification from there. Then I would like so this is the one project where we have developed a polymeric dendronized technology for the in vivo delivery of siRNA. Now, this is the one where we have developed a dendrimer templated technology. This is entirely different and very interesting too. For example, here we have used hyaluronic acid. Albumin is very straightforward, simpler polymer, is innate to our body. Our body got albumin in like major composition is there in our body, right? It is FDA approved, already a product of albumin of paclitaxel. It's already there in the market in the name of Abraxane. That's already there. It's good. It is said that it's good to take the FDA approved versions. So that's the reason we have taken it. But the problem with using albumin for the delivery of siRNA is that they are big negative charge. You cannot load siRNA into it. So here we resolved this issue. But still, interestingly, several investigators have tried to deliver siRNA using albumin, but the transfection or the loading was very less, 20%. Why this 20% was there? Because albumin too got certain amine groups, but our objective was to enhance it. So we have employed this dendritic technology. It's very smart. So first take the dendrimer, which are positive charge, mix it with the siRNA. It will form a complex again, which is positively charged. Load this positively charged dendrimer siRNA complex with negatively charged albumin, which literally happens very effectively because of this positive negative interaction. Following this, add some stabilizers, get this one pot again. Okay, so it's again mix and deliver type. So mix A, add this complex to B, which is albumin, get it done, add stabilizer, and then you are good to inject. Okay, now this is how we first we optimize. Okay, how much dendrimer is being required to effectively load. Okay, so we determined the binding efficiency and we found that zero point 0.5 dendrimers to siRNA ratio is sufficient to form the complex, good for the good for its loading inside the albumin, negatively charged. Okay, so this is the one where zeta potential, after some time we found insignificant decrease. So we have strategically came up with a protocol to mix dendrimer with siRNA. Dendrimer here used as a template. Now this dendrimer siRNA template is being loaded inside the albumin to develop nanoparticles of size. Here the size was pretty small, less than 100 nanometer. Okay, and the encapsulation efficiency, you can see if you try to load siRNA in conventional albumin nanoparticle, it's like less than 20%. We validated it in our lab, but when we apply this templated technology, it goes all the way closer to 75 to 80%. Anyway, they are pretty robust, stable nanoparticle. And here the important thing is that if you compare with conventional SIANP is a conventional uh, um siRNA nanoparticle. So if you see under acidic environment, this DT-SINP, they are more sensitive. See here it's like its size went from 40 nanometer to around 120 nanometer, and its size from 40 nanometer to 60 nanometer. So the proportionate increase in size, the proportionate increase in positivity, or the Z average was found to be significantly higher in case of dendrimer templated version. And again, if you ascribe this, if you want to ascribe this, this is because of the incorporation of the dendrimer template. That is exactly bringing up this change of higher encapsulation and higher proton sponge effect. Serum stability also we have assessed, and we found that they are pretty stable in serum. As such, they do not. So what happens? Why serum aggregation happens? Because uh, albumin, uh, you know, these proteins are there. Circulatory proteins are there in our blood. So whenever the particles of say positive charge, they come in contact with them, they undergo interaction, ultimately leading to the build up in their particle size. But here in this case, there's albumin. Albumin is innate, biocompatible, known to the body. When it comes in contact with the other proteins, which are again negatively charged, so they don't interact. So that's the reason you can see their particle size even after 24-hour was pretty much same, stable. In context to their effect of pH, I have already crosstalk that they are pH sensitive. Okay, this is the interesting aspect. So if you mix this naked siRNA without serum in negative environment, they are stable, but when you mix it with positive, in less than 1 hour time they degrade. This is about the conventional albumin nanoparticle. They are also not stable. So that means here loading was there, but the encapsulation was peripheral, facade, surface, topographical. On the other hand, in case of this dendrimer templated ones, they were found to retain the stability of this siRNA for up to 24 hours. We have tested it and they were stable. What does that mean? That means that they are retaining it inside the interior crevices. Okay. All right. So this is one where we have tested. So in this investigation, we have tagged the surface of albumin nanoparticle. We have not used the albumin as such. We have substituted around 15% albumin with the CRGD truncated albumin, and this is for targeting the diabetic nephropathy. Okay, so our objective was to deliver HDAC4 siRNA into the podocytes. So these basically these cells are the podocytes, renal podocytes. Okay, and basically this is the diabetic nephropathy in vitro model we have developed in our lab. So basically these are the cells which have been first treated with high glucose, so diabetic, and then for a period of time to develop diabetic nephropathy in vitro. So now what happened, if you talk about the FAM siRNA, you can see them, they are inside the cell but not inside the endosome. But when you talk about the DT-SINP, this is the one. So plain siRNA, they doesn't go inside the cell. So here you can see no FAM fluorescence, no fluorescence of siRNA observed until 12 hour. No. Similarly, in naked siRNA, no. In albumin conventional nanoparticle, no uptake. But in case of targeted, that is a integrin targeted one, you can see them inside the cell in 8 hours. And here, what we did, we first label the endosomal compartment by using lysotracker dye. So lysotracker dye is red one. So this red zones represents endosome. Now our objective was to see whether this fluorescence is exactly in the endosome or not. If it is in the endosome, this infers that the uptake is via receptor mediated endocytosis, and we could clearly see that it is leading to yellow fluorescence after developing the merged image. And this confirms that in fact in 6 hours they are inside the endosome, and not that much. After 12 hours we could see this fluorescence dislocating from the endosome to the cytosolic compartment. What this infers? This infers between 6 to 12 hours these endosomes are getting ruptured, followed by release of this FAM labeled siRNA into the cytosol. So this is the one that infers that whatever we have developed, what DT-SINP nanoparticle, they are capable of undergoing receptor mediated endocytosis as well as capable of releasing it inside the cytosolic compartment. And then we tested if it is releasing in the cytosol, whether it is leading to the silencing of HDAC4, because HDAC4 is the one that is ultimately overexpressed in diabetes nephropathy. So if a person is suffering from diabetes, eventually if it is like for a longer period of time, this HDAC4 overexpression takes place in the podocytes, renal podocytes. What they do, they increases the TNF alpha, necrotic factor, they increases the apoptotic gene. So what happened, they lead to the subsequent death of the podocytes. So that's the reason if you see the histopathology of a diabetic patient, okay, or a diabetic mice or animal, eventually after several years, if you do the sectioning, you can see the podocytes got effaced. Effaced means they will go far from each other. So if they are going far, then more space for the excretion, and that is the reason albuminuria is there in diabetic nephropathic patient. Okay, so we found that it could really silence the HDAC4 siRNA, and this is the one which I was just referring. So if you see, this is the control. You can see here that they are having a well placed structure. This is the podocytes one, while here you can see they are effaced. From here they are broken. So what's happening here, this process, this event is called podocyte effacement. So now this is the control healthy mice, this is the diabetic nephropathy one. You can see here this podocytes, this uniform arrangement of podocytes got disturbed in DN mice. This is the, see here, it is broken. So this is the event which is called podocyte effacement, and that's the reason higher albumin area is there in the, this is the albumin, urinary albumin secretion, which is very high there, is because of this. In case of naked siRNA is the same, because it is not effective. In case of the DT-siNP, you can see it is having significant curative role, reversal role. Okay, it is trying to make the podocytes healthy, trying to reduce the urgency, right? Now, it's like 12:33. Okay, so let me summarize my presentation. So in this investigation, I have discussed you about dendrimer templated approach, which we have developed for the delivery of siRNA therapeutics with high entrapment efficiency using some anionic polymer. And the two which are the FDA approved ones like hyaluronic acid and albumin, which is a very simpler technology, just one pot simple mix and deliver, which offers serum stability, in vivo protection, and also lead cytosolic delivery, okay, by escaping from the endosome. We have tested in cancer and podocyte, but the avenues are endless. So it is proposed that this is a model concept that can be explored further for other RNAi therapeutics as well. Maybe for plasmid, maybe for microRNAs, or maybe for some other proteins, peptides too. You can combine the RNA therapeutics with a drug, or the drug combination can be explored, that too for other diseases like arthritis. Okay, and the future node is that years of research has been done, and today we are having a greater understanding of the siRNA therapies. Okay, as evidenced by commercial launch of two siRNA therapeutics on Patro and Gilvary, which is currently available and FDA approved one. And the good thing is that several companies have came forward. For example, Alnilam, as per my recent reading from Economic Times, Alnilam have just now announced their partnership with Regeneron Pharmaceuticals, that they are going to jointly deliver the siRNA therapies for the treatment of CNS and liver disorders. And I'm happy to share that two new siRNA products called Lumacida and Inclisiran have been submitted for FDA nod, and finger crossed that it should go through. If that goes through, then probably we will have two more products in the line in the studio for siRNA therapy. And plus several siRNA therapeutics are under clinical trial and more are in developmental stage. And very recently in COVID pandemic, Alnilam and Veer Pharmaceuticals have also announced that they are literally working on a joint venture that is trying to offer gene therapy using siRNA to treat the COVID-19 severity. And on top of it, there are several other companies who have done massive investment like Dicerna, Eli Lilly, Johnson and Johnson, Amgen, AstraZeneca. These are some of the players which have done like massive investment in in developing siRNA therapeutics. So this is an emerging field, and the medical field is literally having huge hope from this. Several young scientists are here. I'm sure some of you must recognize this gentleman. He is Sir James Dolly Watson, and I'm sure you all know him from his discovery of structure of DNA along with his peers, Francis Crick and Rosalind Franklin. Okay, they jointly discovered the structure of DNA, and followed by this in 1962, they got the Nobel Prize of Physiology and Medicine too. In his recent lecture during 111 SUSTEC conference in Shenzhen, China, Sir James Watson, he tweeted and announced also from the desk podium that, long back there was a time when we humans used to think that our fate lies in our stars, but he said that now there has been a significant and paradigm shift in the thought process. Now he said we should shift our thinking from stars, and he said that by now we know in a larger measure that our fates are also in our genes. And that's the reason he said to to support gene therapy. These are some of my lab members who are directly involved in this work: Nidhi, Vishaka, Kaushik, Prof. Kiran Kalia. Prof. Kiran Kalia is the one who take care of our biotechnology related work. Nidhi Rawal, she did her PhD in one of the product. She is currently doing postdoc from UK in Angelina Ruskin. Dr. Rahul, he was a postdoc in my lab, and currently he is working as a faculty. On top of it, Nilima, Dhaneshwar, Surya, and one more new candidate recently joined, Nupur. She is also working on this project. I thank every one of them for their hard work, working round the clock in the lab. You can see them in the lab, like, late night and early weekends. They are here. I would also like to take a small moment to thank our funding agencies, including Department of Pharmaceuticals, ICMR, SERB, MOSTI, British Council, of course, for Troika, and our professional collaborations from different places. And with this, I would like to give myself pause. Thank you very much for your attention. May you have a very fruitful journey, learning experience with this conference. Thank you very much.
Thank you, sir. Thank you, Dr. Rakesh Tekade, sir. I request Dr. H.S. Mahajan, sir, to conclude this session. Please, sir.
Okay. Thank you, Dr. Tekade. Kindly accept my congratulations as well as the compliments for your wonderful session on dendritic technology as a next generation sRNA delivery model. Dr. Tekade discussed during his talk the importance of sRNA therapeutics, and he has taken to us to the central dogma of life and how there is a conventional APIs are affecting the current treatment strategies are there, and how sRNA therapeutic, or how we can think about the RNA as a next generation therapeutics is that. Dr. Tekade talk about some problems with the sRNA delivery. Currently, what are the problems majorly to be occurring in the development of this sRNA-based therapeutics, that is in vitro, in vivo disconnect. He mentioned about that, especially he mentioned about the short elimination half-life and anionic nature of the this genetic matrix. That will be some problem in a translation from lab to the actually in view. And he very well explained with us about the biostabilization of this genetic materials using some hybridized dendritic technology by using some dendritic polymers and their property of protonation in order to load these genetic materials in these techniques are there. And he come up with some novel technologies using albumin and hyaluronic acid for the delivery of these genetic materials in order to overcome what are the problems that are associated with the currently available transfixion technologies are there. And Dr. Tekade, in a very simple language, explain about what kind of the methodology he developed and how simple it is to carry out in a very using a very available infrastructure facilities in a single step process is that. He talked about what is the future of this technology, and after attending his session, we are thinking that definitely, but the RNA therapeutics are there. That will be the future therapeutics, and most of the people may not be used the APIs for the treatment is that. And Dr. Tekade during his session giving the focus of this gene therapy, and major work is going on in a cancer studies are there. 70% of the research work is going on in a cancer diseases there, and you might all aware about the what are the complications or limitations of the currently available chemotherapy of the cancer. Definitely, in the coming future, we'll have some better treatment for the management of the cancer is there. Dr. Tekade also given some market growth of this RNA-based technologies or gene therapy, and the current CAGR in 2020 is about 7.7%. That shows the what is the potential of this market is there, and definitely there will be some lot of job opportunities as well as the research opportunities into this area is there. At the end, while summarizing, Dr. Tekade given some works which are going on at a commercial level is that most of the companies are doing a lot of works, and as far as the India is concerned, the Biocon is one of the leading biological waste company. We hope by doing a lot of research work in this area, will have some cost-effective therapeutics, genetic therapeutics for the management of the certain diseases which are not currently not having the good management strategies available with us recently. Before concluding this session, uh, I wish to ask one question to Dr. Tekade. Is there any regulations are there for the getting the approval for these genetic materials, and how they are differ from the what is the currently available regulations for the APIs and new chemical entities?
Yes. So first, let me explain you for this siRNA therapeutics. No separate regulation is required. The same regulation which are there for transumab or any biological, the same applies here. The only thing which the scientific fraternity is worrying at the moment is about the safety and safety of this basically. So long term safety data is required for this, which currently they don't have. And so it doesn't require any separate set of regulation to be followed. The ones which are already existing has to be followed. All the method and boxes that need to be assured is related to its safety and efficacy, so that has to be taken care.
Sir, there is one more question. I think every attendee must wish to ask the same question. What could be the attempts needs to be made by the scientist in order to reduce the cost of these products?
So the question is how we can reduce the cost of this technology. Is that correct?
Yes, sir.
Okay. Yes. So as I said, this is actually very valid and very pertinent question. Thanks for asking and bringing it forward. Actually, this siRNAs, they are way cheaper. If you talk about 1 gram of melphalan, it will cost like more than 50,000. That too concerning its IC50 is in micromolar range. In context to siRNA, they, if you talk about the cost of therapy, it will come very cheap, very cheaper as compared to that of the drug. Okay, so currently the only problem with siRNA is it lies in its delivery modality, which is missing. How to deliver it, we don't know. Okay, so that's the problem, and that's the reason people are trying to give, like, several higher fold, because they are considering that if we deliver 100 picomole, if we deliver 100 picomole, only 2 picomole will go at the site of action, 98 picomole will get degraded. So we are seeing the literature, or we are seeing the clinical trial that is working at a superfluously higher level of siRNA, which is not required. If we have a very fit to purpose delivery modality, we just need to give 3 to 5 picomole of siRNA. If we can attain that, then it will bring the cost of therapy even cheaper than that of the uh, currently contemporary therapeutics. One more thing I would like to add, for example, I said Givlari is in market for the treatment of acute hepatic porphyria, and it was found like very effective. It has given fast track FDA approval without having a concrete safety data. FDA gave approval because there was no other alternative medicine. But if you hear about its regimen, you will be surprised. It is to be given once a month for 6 months. So the frequency of dosing is too less. Plus, this is the cost what we are discussing now. When we do not have an precise mode of its delivery. Imagine if our scientific fraternity down the line comes up with certain very fit to purpose delivery modality for siRNA, the cost will go like 98-fold lower at least. So uh, talking about their cost of therapy, it will be way cheaper. For example, um, just a second. For example, here, if you talk about uh, drug therapy also, I must say, right? For example, conventional chemotherapy, right? If you see how compromised Medicare system we are currently witnessing, I must say, right? For example, if you see a cancer patient, okay, say fluorouracil, if a cancer patient is on fluorouracil therapy, he has to be kept on infusion. Why? Because of the short half life of 35 to 45 minutes. So it has to be given as an infusion. If he goes to washroom, he has to carry that infusion set with them, because it has to be given. So now why that's happening? Because we do not have a exact precise delivery modality for fluorouracil as well. So for fluorouracil also we do not have. So now how much percentage of the drug delivered or administered to the patient goes to the site of action is just 1 to 2%. There also we are losing significant portion. So the questions still remain open for both. In conventional drug therapy also, we need certain very precise delivery modality, targeted ones that can exactly deliver it at the site of action. So if we can exactly capable of delivering this 2% drug to the site of action, we need not to give 98% extra. So this
Sorry
is
to
one thing.
Dr. Rakesh, sorry to interrupt you, but Dr. Anand is waiting in the waiting room. We shall have
Okay,
conversation later.
sure, sure.
I request Dr. Hitendra Mahajan sir to have personal words with Dr. Rakesh, and Dr. Rakesh is very helping, I think. So I would like to conclude your session, Dr. Rakesh. Sorry, extremely sorry for
Sure,
interruption.
sure. No,
I
no,
remember
Prashant.
a quote of Marie Curie. I think there are very few scientists in the field of medicine and physics and chemistry. So Marie Curie received Nobel Prize in physics and chemistry. So I think he invented radium and polonium also. So there is one
Yes.
quote: "Have no fear of perfection; you will never reach it." So Dr. Rakesh, you proved yourself in today's session. Um, you have very young and dynamic team at your end at NIPER Ahmedabad. I would like to say thanks to Dr. Hitendra Mahajan sir, session chair of this particular session, for sparing his valuable time. Thank you, Mahajan sir. Thank you very much. I would like to say thanks to Dr. Rakesh Tekade sir for sparing his valuable time. Thank you very much, sir. Thank you.
Pleasure.
Sir, with your kind permission, shall I leave the studio?
Yes.
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 Thank
Thank
you,
you.
sir. Thank you.
Thank you. Have a
Thank
good
you.
day.
Thank you. Thank you.